Let's start. So our first speaker is going to be Ashby Kissoondoyal. He is a PhD student in Dorota Crawford's lab in York University. So he's doing very interesting work on how abnormal Ptg2 signaling is linked to autism spectrum disorders. And if you want to learn more about his research, you can also refer to his paper that's been recently published, and we will add a link to the chat. And so today he's going to talk to us about how the effects of abnormal Ptg2 signaling on the morphology on the dendritic spines and observe differences between male and female mice. So with that, please, Ashby, you can go ahead.
All right, thank you, Katya. So again, yeah, I'm Ashby, and today I'm going to reiterate, talking a little bit about a project in which we quantify dendritic and dendritic spine morphology in a COX-2 knock-in mouse model. So, there we go. So before I get into the details of the particular study, I'm just going to talk a little bit about what our lab does in general. So we primarily study the effects of lipid signaling on neurodevelopmental disorders, particularly focusing on autism spectrum disorders, or I'll just call it ASD for short. The prevalence of ASD has been increasing at quite an alarming rate over the past few decades, actually. And as you can kind of see here, in 2014, this rate was 1 in 59, so very, very high. Another interesting fact about ASD is that males are at a much higher risk of being diagnosed with ASD, being roughly 4 times more likely than females. So the current prevailing opinion on ASD is that ASD is not solely a result of genetic factors, nor solely a result of environmental factors, but actually a combination of factors from both types. And our lab specifically looks at abnormal lipid signaling because lipid signaling can actually be affected by both of these environmental and these genetic factors. So a little bit more on lipids here. They are a major component of the brain, making up the majority of the brain by weight, and they serve as a cache of signaling molecules that are released when they're required. The lipid we focus on in our lab is the molecule prostaglandin E2, or PG2 for short, and it's one of the more prominent signaling lipids within the brain. So we know that PG2 is involved in neuronal development, but also in synaptic plasticity. So on the side here, I've included the PG2 synthesis pathway. Arachidonic acid is converted into PG2 through the action of this Cyclooxygenase 2, or COX-2 enzyme for short. What's interesting about this pathway in particular is that an assortment of different environmental factors can impact it. Things like air pollution, NSAIDs like acetylsalicylic acid that everybody's very familiar with, as well as other consumer products and over-the-counter drugs can impact different parts of this pathway, either increasing or decreasing overall levels of PG2 in the brain. Our lab in particular is interested in the downstream effects that these increases and these decreases in PG2 can have. To study this, we use an in vitro cell model that's exposed to PG2, as well as 2 in vivo mouse models, one in which we expose pregnant dames to PG2 through injection and study their offspring, which serves as our high PG2 mouse model, as well as another model in which we reduce the levels of COX-2, our COX-2 knock-in mouse model, which serves as our low model. So downstream, we've seen that changes, these ups and downs in PG2 can cause changes in gene expression that cause these functional changes in things like migration, proliferation, differentiation, and synaptic plasticity. More interestingly, I think, we've also seen behavioral manifestations of these ups and downs in affected mice, which have shown social deficits as well as repetitive and anxious behaviors, all things that in humans have been used to diagnose ASD. For the purposes of this study, though, I'm just going to be talking about this COX-2 knock-in mouse model. So the primary objective for this study was to investigate differences in dendritic and dendritic spine development in wild-type and COX-2 knock-in male and female mice in the cerebellums at post until day 25, with the hypothesis that a COX-2 knock-in would disrupt the normal development of these dendrites in their spines, resulting in abnormal morphology and of course further that these effects would be modulated by the sex of the mouse. To examine dendritic morphology, we used a technique called Golgi-COX staining. And so with this technique, we can specifically actually stain neurons and their dendrites, allowing us to see them in very detailed ways. For this presentation, I'll be talking about findings in dendritic arborization, which we measured using shoal analysis, axonal loop formation, which I'll talk a little bit more about later, dendritic spine density, which is just the number of spines per length of dendrite, and lastly, spine shape and morphology, which we classified based on length and width measurements, and then looked at the odds of each spine being a mature shape, so to simplify, just mushrooms, or immature, one of the remainder. So the first trait I'm going to discuss is actually dendritic arborization. So to quantify this, we use a technique called shoal analysis. For those of you who are unfamiliar with this technique, a program will create or draw in circles, concentric circles that are 10 micrometers apart from the center of the soma outwards. And the number of intersections that the extensions of a neuron make with these circles give us an idea of how branchy a neuron is at that distance from the soma. Looking at the graph here, we have the distance from the soma along the x-axis, as well as the number of intersections these neurites make with the circles along the y-axis. Then we have males in blue, females in red. And to differentiate our wild type and our COX animals, I've used a darker color for the wild types and a lighter shade for the Ptgs2 Knockins. We firstly noticed that there was no actual sex difference in terms of dendritic arborization. However, we saw a dramatic increase in the number of branches of these Ptgs2 animals at this 20 to 40 micrometer range. We actually saw that this was reversed once we got to the 60 micrometer length, with Ptgs2 animals having fewer branches. And I think it's important to note that while there's no significance beyond this point, we actually did continue to see, in terms of the numbers, that Ptgs2 animals had fewer branches. So changes like this in dendritic arborization can actually have an impact on overall brain shape, as well as in the formation of synaptic connections. And these types of changes are actually usually a good sign that there's some kind of dysfunction in axonal navigation. Axonal looping. So when we think about neurons and how they look when we stain our slices, we normally think of neurons looking like this, with extensions that curve and turn, yes, but that tend not to turn back on themselves or their dendrites very often. When we were looking through our slides, we noticed that while the majority of them, yes, did tend to look like this, we actually found a decent amount of neurons with dendrites that would loop back on themselves or form these what we call axonal loops. In order to kind of quantify what was going on here, we measured the exterior angle of all of these dendrites. And when we plot all of these measurements with turning angle on the y-axis and then separated out by condition on the x-axis, again with males in blue and females in red, you can see the distribution of these turning angles. What we did then was we classified any dendrite with an exterior angle greater than 270 degrees, so when it turns beyond that 270 degrees, as looping. And when you separate these points out based on that 270 degree distinction, just looking at the top, for example, you can visibly see that there's a difference in the spread of these points. To statistically examine this distinction, we used logistic regression, looking at the odds that a random dendrite picked out of this pool would be classified as looping or non-looping, given its condition and given the sex as well. So matching what we see visually, we can see that sex had no effect on the odds of a dendrite forming a loop, but that when we look at Ptgs2 animals, that they were highly more likely to form a loop. The degree of this increase in the Ptgs2 mice we found suggests a dysregulation in kind of axonal pathfinding again, and potentially even more so, cytoskeletal dysregulation, both affecting, again, axonal migration and ultimately the formation of synaptic connection in these mice. when we moved on to look at dendritic spine, the first thing we examined was dendritic spine density, in which we measured the number of spines per micrometer of dendrite on the y-axis here, and then again we have condition on the x-axis split into males in blue and females in red. We saw a surprisingly great difference in spine density between just our wild-type males and females, with wild-type males having a significantly greater spine density than females. Interestingly, in our COX-2 animals, we no longer saw this difference. So given that these changes in dendritic spine density overall are often associated with different neurological, neurodevelopmental disorders, this loss of a sex difference, while it's no longer there, may actually correspond to, we think, larger scale physiological and behavioral changes. Now, looking at density is great, but we know that dendritic spines, they tend to have different shapes that correspond to their function, right? So dendritic spines, they change shape throughout development, with more mature spines typically being mushroom-like and contributing to synaptic connections, whereas less mature spines tend to be more transient in their shapes and tend to have these thin, these stubby, these filopodia-like shapes, and are in general less involved in these connections. So we classify dendritic spines into shapes based on length and width measurements, and then we use, again, a logistic regression to examine if within our conditions, any of these conditions or these sexes would be more or less likely to have mature or immature spines. What we found was that wild-type males were actually more likely to have mature spines, whereas females were more likely to have these immature spines. We saw a complete reversal in the COX-2 animals, with COX-2 males more likely to have immature spines and COX-2 females more likely to have these mature spines. So spine profiles, which is to say the proportion of different spine shapes at different ages to one another, are often again associated with different states of neurodevelopmental disorders as well as neurological disorders, suggesting that these COX-2 mice, given the dramatic switch from the wild-type condition, may also exhibit physiological or behavioral abnormalities again. To conclude, I've included the findings I discussed today into a table with the measures over here, as well as the kind of a summary of the effects we saw. So with regards to dendritic outgrowth, our findings suggest to us that the COX-2 knock-in affects axonal pathfinding and potentially results in some kind of cytoskeletal dysregulation, with the implication, of course, that these mice may be improperly forming connections within their cerebellums. Our spine findings have shown us that there are sex-specific effects of this COX-2 knock-in, resulting in different spine profiles in these mice. So these spine profiles may correspond to improper formation of synaptic connections throughout development. And to reiterate again, in literature, these differences in spine profiles are often correlated with neurodevelopmental disorders and neurological disorders. Given that our study has specifically examined the cerebellum, it seems likely to us that these changes in dendritic outgrowth and these changes in spine profile suggest that these mice, these COX-2 mice, may have deficits in cerebellar-related behaviors, with specific sex differences, of course, in the spines. To wrap up here, I'd like to, of course, thank Dr. Crawford, as well as the rest of the Crawford lab for their support, as well as these two facilities here for helping with the slicing, the staining, and the imaging of our slides. And of course, lastly, our funding agencies as well. I think at this point, I'd be more than happy to take any questions anybody might have.
Ashby, thank you so much for your talk. It's very, very interesting and very comprehensive study. So I think we might have a time for one question. So far I don't see any questions in Q&A box, but I have a question with regard to, so what do you think underlies this sex differences in some differences in morphology, but not the others, specifically the difference in the spines, but not so much in the length of the branches and branching itself?
Yeah, so I think, so from what we've seen so far, it just may be the trafficking of different elements. So Pg2 itself is synthesized and can be synthesized in a very sex-dependent way, and its localization, as well as what it acts upon, may be just regulated differently within these axons, either to growth cones or other places along that dendrite as well. So I think that might be part of the reasoning.
Mm-hmm.
Mm-hmm.
Yeah, that seems plausible.
Mm-hmm.
Okay.
As a question, do we still have time? Oh, how to understand the axonal looping? Like what happens there?
Yeah, so that one, so from what we've seen is that typically when you have very dysregulated actin polymerization, or is kind of one reason why you might have these looping occurring. Another one is that there might be the inability or improper ability of these growth cones to receive proper navigation cues. So we're not sure, based on our findings, of course, which one it is. I suspect anyways, that it might have more to do with cytoskeletal dysregulation, given some other findings we have with regards to Pg2. But I think, so I think that's probably what's going on there.
Thanks.
Mm-hmm.
Okay, thank you so much, Ashby. And if there are more questions, you can